Hello, everyone. We're giving everyone a few seconds to join from the waiting room. Always great to be here today. I'm Sabrina Paganoni. I'm really happy to be here with Dr. Sukovic and Dr. Berry from the HEALEY Center, as well as Catherine Small from our patient navigation team. I see people are still joining from the waiting room. Really great to be here every week. We really love to provide updates and have an open conversation with the community, and we always love to receive your questions, so feel free to type them in the Q&A box. Next slide. And this is, as always, our, our information for joining our ALS Link, which is our newsletter, as well as the webinar registration. Feel free to share all of these with friends and relatives. Next slide. I really want to thank a participant on this weekly webinar who sent us a great message last week after we had 2 successful webinars with our guest speakers, Dr. Maragakis and Dr. Rofstein. The recordings are still available in case you missed them, and it was really wonderful to hear from participants that they really appreciated all the explanation of the fantastic science, new biomarkers, lots of new hope. Definitely our guest speakers really gave us very recent updates about the latest in biomarkers and the importance of measuring biomarkers, especially in the spinal fluid. And so feel free to go back to those recordings if you want to learn more. Next slide. And again, we continue to partner with other groups such as the NILS Consortium, the ALS Association, to help with provide educational webinars. So certainly we continue the series on Thursday afternoons, and we record them on our website. But we're very much excited to share information about other webinars. The NILS Consortium will have a webinar with Dr. Maragakis, who also was here a couple of weeks ago, on the importance of lumbar puncture and CSF biomarkers and why they're so important for drug development in ALS. So again, this topic I think is very important given the latest in biomarker development. So feel free to join Dr. Maragakis by using the link or the QR code that's on the slide. Next slide. And we're really excited to share our latest metrics on this webinar, but also in general, the patient navigation team, Catherine and Alison. Alison couldn't be here today, but they're one or both of them is generally on this webinar and is always available to answer questions even outside of the webinars. I know many of you send them questions or messages, and they are always available to connect you with sites and research opportunities or resources. And we're really excited that thanks to their help and their coordination, we've been able to really develop a number of central resources, brochures, materials that you can download from our website or ask them to send to you. And really, we're excited that literally thousands of people have been watching these recordings from the Thursday webinars or our drug science webinars. And we really want to continue to have guest speakers to continue to bring you new information, new topics, always the latest. And that's why we actually invited Dr. Berry tonight to join us to talk about the biomarkers again, which is really a hot topic right now, as well as the importance of lumbar punctures. Next slide. So recently we had a fantastic webinar. This was actually on Monday, this past Monday, to talk about the science supporting Regimen F, the new regimen, which just started enrollment recently. In fact, we have a press release also. Everything is posted on our website. So thank you, Catherine, for sharing all the links on the slides and the QR codes. We also post the slides from these webinars on our website. So if it's hard for you to write down or catch the QR code during the live presentation, feel free to download the slides, and you can always use them to go back to the original, to the other recordings or materials. Next slide. And so right now, again, we are testing multiple drugs. As you know, some of them have already completed enrollment and had some results. Regimen E recently completed enrollment and is still ongoing. Regimen F actually just started enroll the first few participants, and Regimen G will come online very soon. Next slide. Right now, in terms of Regimen F, that's the one regimen that's enrolling right now. But again, Regimen G will follow soon. So that's enrolling at 23 sites, and we continue to activate sites. So as you know, we have a network of about 70 sites. We enroll sites on a rolling basis. And so right now we have 23 sites that are approved to enroll participants in Regimen F. So if you're interested in enrolling right now for Regimen F, again, those are the 23 sites that are active right now. You can always find their contact information on our website, or please call Catherine for any additional questions. Next slide. And Catherine and the team have also prepared a number of resources that are downloadable from our website. We have brochures about the trial in general, and also in particular about Regimen F, which is the regimen that's enrolling right now. And because biomarkers are so important and lumbar punctures are so important, we also have additional brochures and information available on our website about these procedures and topics in particular. And we're also working on adding more resources to our website. Next slide. And again, these additional images of the lumbar puncture to really give us more information, give everyone more information if you have questions. Next slide. But it's also great to have a live speaker who can answer your questions right now live. So in addition to the brochures, we really wanted you to hear from an expert who has been doing, I don't know, thousands of lumbar punctures perhaps, or I don't know, tens of thousands. And so Dr. Berry really is a leader in many things, including really an expert in on biomarkers and lumbar punctures. So I'll give the mic to you, and we can talk a little bit about that. And obviously we'll wait for questions from the audience.
Yeah, absolutely. Thank you so much, Sabrina. It's always fun to be here. It's such a joy. The first thing I would say is that, I think what Jeff Rothstein and Nicholas Maragakis talked about is really the importance of having CSF so that we can check biomarkers in the CSF. CSF stands for cerebrospinal fluid. This is the fluid that bathes the brain and the spinal cord. It's actually, you should think of the spinal fluid as a slow-moving river, not a lake. So it's, we sort of make this every day. You make about a soda can's worth of spinal fluid every day. It's made in the brain. It kind of flows down along the spinal cord and then back up to the brain where it's sort of retaken up. So it's just filtered from the blood and then put back into the blood. The reason that, people who are in the lab doing biomarker development, like Nicholas and like Jeff and like so many others in our ALS community, are so kind of enthusiastic about having spinal fluid to analyze is that this is the fluid that is in direct contact with the motor neurons. It's really bathing the motor neuron. So if something's going wrong at the motor neuron level, those cells are sending signals of that directly into the spinal fluid. The spinal fluid it's less complex than blood. Blood has cells, it's got clotting factors, so it's a really complex fluid. Spinal fluid is much simpler. It's a clear fluid, and it has fewer proteins in it. It has less abundant proteins, so we can kind of really look for these important biomarkers there. Some of the science that's coming out of CSF biomarkers right now is remarkable. You heard from Jeff Rothstein and Nicholas Maragakis about these markers of what we call TDP-43 pathology, the sort of the central problem of ALS. So really, really exciting. Now, there are lots and lots of biomarkers discovered and as yet undiscovered that we can look at in the spinal fluid. And it has had the central role all along. And I think it's really important that we talk about not only that it's so important and how we analyze the fluid, but how do we give it? How do we get it? And what's that procedure like? So the way that we get spinal fluid is by doing what we call a lumbar puncture. Sometimes people call that a spinal tap. And essentially what that means is we go down to the lower part of the back and we insert a needle into the spinal fluid. So the reason we go to the low back is because actually the spinal cord ends somewhere around your belly button or a little bit above. And below that, there's no spinal cord anymore. What it is just nerves that are floating in this fluid. And so there isn't a risk of, injuring the nervous system in that way. As far as sort of like injuring the spinal cord directly. So it's a very safe procedure, actually. We're just like when you draw blood, you just sort of get to the right spot and the blood comes out. Same thing with the spinal fluid. The procedure is typically done in the office. There are two major ways that we do it. One way, people sit up and they sort of punch over so that it opens the spaces down in the lower back and we can get to the right spot. Another way, people lie down on a table, like an examining table, or a bed. And it can be done either way. Most of that is just the practicality. Sometimes there are reasons to have people lying down versus sitting up, but most of the time for research, when we're just trying to look at proteins, it doesn't make a big difference. We can do it either way. And that's a conversation that you should have with if you're undergoing a lumbar puncture, you can have that conversation with the person who's doing it. What position should we do this in? And 99.9 times, it's a conversation, and you can kind of come to a shared decision about that. We use numbing medication, so there is a little pinch with the numbing medication, and then some pressure with the needle, with the spinal needle. Unlike blood, we can't just draw the spinal fluid out by sort of sucking it out. We have to just kind of let it come at its own rate, and so it can take some time for it to come out. But once everything is in place and we're just having that fluid come out, it's a pretty quiet time. There isn't anything in particular that we do after a lumbar puncture that's critically important, but we do usually have people lie flat for somewhere between 30 minutes and an hour, just to make sure that they everything is that they're doing well, that everything is sort of stable before they get up and go. We tell people not to exercise heavily that day, and we tell people to drink plenty of fluid and also to drink their normal amount of caffeine. And all of this is really aimed at trying to reduce what is maybe the most common risk of a lumbar puncture, which is that it can cause a headache. So when we withdraw the spinal fluid, that sometimes can cause a headache. Now, then this is really the major risk of doing a lumbar puncture or spinal tap. The things that we can do to try to reduce the risk are making sure that people stay hydrated, that they drink at least their normal amount of caffeine, because we don't want people to go into caffeine withdrawal, which can also induce a headache. And having people, sort of lie flat for a while after the procedure. But the biggest thing that we know, and we've published on, but lots of groups have published on as well, is that we can use a needle called an atraumatic needle, and it just does a little bit less damage to the tissue as it goes in, and makes for a smoother lumbar puncture experience and can reduce the amount of headaches that we see pretty substantially. So that headache risk is somewhere around 10%. And of those, most are not major headaches. Clearly, if someone has a headache, they should get back in touch with the person who did the lumbar puncture, and there are pretty effective medications we can use if someone needs something stronger than a Tylenol or an Advil. And in rare cases, there is a procedure we do called a blood patch, where we draw blood from a person's own arm and then put that back where we did the lumbar puncture, and it sort of stops any leak of fluid there and helps with headache. So that's kind of the whole thing. That's very uncommon. I mean, we do it a few times. It's very, very uncommon. I would say most of the time when we do a lumbar puncture, people's reaction is something along the lines of, "Oh, that's all." It usually goes quite well. What we get from it is clear fluid. We can get somewhere between 5 and 20 cc's of cubic centimeters, milliliters of fluid. And then we can store that. And I ran a large, along with others, in our community, and many participants ran a large longitudinal CSF study where people had somewhere between 3 and 7 lumbar punctures every few months about 10 years ago. And we still are using that spinal fluid for really critical studies today.
Thank you. So again, incredibly important to get the spinal fluid to be able to have it for research. And again, it's a procedure that's been done, again, many times, and most of the times, it's really essentially a very easy procedure. Again, the risks are really, really limited. Just a question: how long does it take in the body? Does it take the body to replace the CSF that you take with the lumbar puncture?
So probably on the order of a couple hours, something like that. Again, it's just filtered from the blood, and so you make more of it. And the body does pretty much maintain the amount of spinal fluid pretty carefully, so it would be replenished pretty quickly.
Right. So somebody asked, I had trouble in the past with this procedure, and the person who did the lumbar puncture had to stop because they were not able to get any fluid. Is that a reason why?
So, well, the challenge of getting spinal fluid is that we can't see exactly where that, I can't see inside at the same time that we're doing it. And so sometimes the angles just don't line up the way that we would expect. And so it can be hard to get to the right spot. That's honestly the most common reason that we have a problem. It does happen. It's happened to me as doing the procedure that I couldn't get spinal fluid from somebody. And sometimes that's just positioning. Occasionally someone has, we find someone who has really severe arthritis. So your vertebrae are just joints, it's joints between your vertebrae, and they can get arthritis and get a little, the space that we're going through can be a little bit smaller. Most of the time it's positioning, honestly. And so even when people have had a lumbar puncture attempt where they weren't able to give spinal fluid, it can be worth trying again. Again, I referenced the study where we did longitudinal lumbar punctures, and every now and again, there would be someone who had everything went smoothly twice, and then one time I just couldn't get to the right spot. It was rare, but and I think it's just something about positioning. So it's often
question: why is lumbar puncture not usually used in the diagnosis of ALS?
So ALS is a diagnosis of exclusion, which means that we need, there are a few tests we do, like an EMG, that give us really signals that are consistent with ALS and abnormal. But most of the testing we do looks essentially normal in ALS. That's true of an MRI. We're doing an MRI of the brain or the spinal cord to rule out other diagnoses, not to find some signal of ALS. A spinal tap or lumbar puncture is the same in that it should look relatively normal in ALS. And so it just often doesn't rise to the level of sort of needing that evidence during the procedure to kind of rule it out. Now, sometimes there are particular cases where a lumbar puncture is important for diagnosis because we're trying to specifically look for another diagnosis and specifically make sure that there's something else that's not going on. That's really when we would do it. If we were looking for an infection, if there was a reason to suspect that there was an infection in the spinal fluid, for example.
Question: who does the LP? Is it a physician who does the procedure or a coordinator? What type of preparation, I guess, or certification do they have? Yeah,
so it's generally a physician or a nurse practitioner or a physician assistant. Those are the kind of 3 categories that I can think of that would be qualified to do a lumbar puncture. Lumbar punctures there's always a training process, observing, doing with close supervision, and then beginning to do them independently. So that's how we learn, and we learn over many years. And but that's, that's really that's the person who's going to be doing it.
Do people need to prepare for study visits any differently if they have an LP visit compared to a standard visit?
Typically, no. I mean, I think, you want to be hydrated when you go into your visit because you're going to give blood anyway, and probably urine, and, just like blood and urine, things flow a little bit more if you're plenty hydrated, but I wouldn't take that to the extreme. I do think, just planning your day so that you don't have a lot of, strenuous physical activity after your lumbar puncture is probably a prudent thing to do. And I will say that if people have a long plane flight or a long car ride, I often advise them after a lumbar puncture, maybe to put that off until the next day, if at all possible. This is not backed by science. My observation is that, when people are kind of, sitting in a position for a long time afterwards or can't get comfortable, that maybe the headache risk is a little higher. Nothing devastating. It's just trying to reduce the headache risk.
Can you drive yourself to and from a visit when you get in a lumbar puncture?
Yeah, you can. I mean, it's, if somebody's driving on the way to the visit, they'll be able to drive on the way from the visit. We don't use a general anesthetic. So there's nothing that sort of makes someone confused. We use a local numbing agent just in the low back. And so, both physically and cognitively, people should be doing okay afterward.
Is it different from an epidural that you get for childbirth, or does it feel the same?
So it's done in in very similar way. I'll do a little terminology here. The dura, called the dura mater, stands for tough mother. The dura mater is the lining that keeps the spinal fluid inside. So if we were to put what an epidural does is it goes to just outside the dura. It doesn't go into where the spinal fluid is, just outside the dura. And we inject medicine there, which then sort of actually does sort of the medicine itself transmits itself into the spinal fluid just by flowing. But the needle should not go into the spinal space. The problem is that, we can't know that perfectly. So lots of times when people get an epidural, actually the needle goes into the spinal space anyway. So it ends up being a very similar procedure. Now, the needle that's used for an epidural is much bigger than the needle that we use for a lumbar puncture. And the size of that needle and the shape of the needle are the biggest determinant of headaches. So for that reason, headaches are probably more common with an epidural.
Okay. There is a concern about the time after the lumbar puncture. So somebody asked, you mentioned laying flat, but the person who's asking said, I have problems with that due to sleep apnea. Is it necessary to lie flat?
So if someone can't lie flat, then we usually have them, rest at a comfortable position for some period of time. And I will also just be candid that there is no real well done research that says that that's necessary at all. In fact, in our lumbar puncture clinic, people just, for the hospital, people just rest until they're comfortable to go.
So you spoke about the importance of biomarkers. Will biomarkers eventually carry more weight in terms of determining if a drug is effective or not than the functional rating scales, or do you think they'll always be together? We'll always need both.
Always is a big word. So what I would say is that we saw last week with the tofersen advisory committee, really strong signal from not the FDA, but advisors to the FDA, that they were willing to accept that a reduction in neurofilament was reasonably likely to signal clinical benefit. And I think that is, the evidence for that is growing and growing. So neurofilament is one biomarker, but there is evidence tying that to disease progression. And so I think that we are beginning to gather evidence that, we can follow neurofilament, and if neurofilament is reduced by a substantial amount, that may indicate clinical improvement. I think that is still not it's still not in lockstep. So we have some drugs that have clinical effect, but don't affect neurofilament. And it may be possible that we could affect neurofilament and not have exactly the same clinical effect. So I think for now, we need both, but we're getting more and more to rely on a biomarker like neurofilament. As we develop more biomarkers, I think we may have even more confidence in them. And if we look at the Alzheimer's field, they really have gone, at least in the diagnostic part of Alzheimer's, they rely on biomarkers a great deal.
Great. Question, this is not about biomarkers, it's more about participation in general. So somebody's saying that is in the open label extension for Regimen E. Another question is, what would, why would somebody want to stay in the open label extension as opposed to stopping that and move into a new regimen? Can you maybe talk about general considerations about this type of decisions?
Yeah, sure. These are really, I think, personal and complicated decisions. So if somebody's been through Regimen E, through the randomized controlled portion of the study and into the open label extension, so they know that they're receiving the study drug at that point. I think for the drugs that we're testing, it's hard to know on an individual basis whether they're being effective because there's so much variability in how the disease progresses. And though we know that some people are faster progressing and some people are slower progressing, day by day or week by week or even month by month, there's a lot of variability even within people. This disease is not a straight line. So it's, I think, hard to know for sure whether a drug is having a benefit or not. I think the part of the reason that people would stay in an open label extension is because it's, they're actually receiving the study drug and have been through a lot to kind of get to that point and are continuing to contribute data on that regimen. At the same time, if somebody said, I've sort of been a part of that and, gone through the randomized controlled trial and been in the open label extension and I want to try something different, we do have a mechanism for coming out of the trial and waiting for a period of time to let the drug wash out and re-enroll in the trial. Sometimes people are ready to try something new or contribute in a different way. The one thing I would say is you do need to still qualify for the trial again. So you might want to just, do a little pre-work to make sure that, you would still fit the criteria as well as you can tell.
Yeah, last question about the lumbar, or actually a couple of questions about the lumbar punctures. So you mentioned the reduction of headaches when we use an automatic needle for the lumbar puncture. Actually, two questions. Number one, do most ALS clinics use this type of automatic needle? And the second question is, how about the trial? Are we going to use that in the trial specifically?
Right. So in the trial, we provide the atraumatic needles for sites to use. And we've also been working with investigators that, if they haven't used them a lot, to make sure that they understand sort of how they work. It's very, very similar, but, we've just been making sure people are comfortable with it. I would say that, 12 years ago, 15 years ago, I don't know, when I started doing that longitudinal spinal fluid study that I talked about, I had never heard of an atraumatic needle. I think most people hadn't. They existed. They were becoming used for some things. This one is sort of personal for me. When I was a kid, my sister had lymphoma cancer, and she used to get spinal taps to give chemotherapy into her spinal fluid. It made her very ill. And so I remember sitting on the couch with her while she was, feeling very sick. Now that's very different because she was getting chemotherapy into the spinal fluid. But I thought, I don't want to do that to people. And so anything I could do to make this procedure feel better to people, I wanted to do. And so we actually worked with anesthesia. This was really Merritt and I worked with anesthesia to figure out all the things that we could do to reduce headache risk. And that was when we learned about the atraumatic needle, which was being used in anesthesia. And then we published a paper about it, but many others haven't. In fact, there have been cost effectiveness studies done about this that show that actually the cost to a hospital is less because if fewer people have headaches, there's fewer medications that have to be given out, and you can do more of these procedures. And so I would say that now departments across the country are beginning to rely more and more on the atraumatic needles for every reason, it's better for people with who are having a lumbar puncture. And there's actually a bottom line benefit for hospitals and departments. So I think everything aligns, and more and more people are using this.
Great. There's a question that has many parts to that, but is the blood-brain barrier preserved in ALS? That's sort of the first part. And then the second part is, if not, can we detect biomarkers in the blood using sensitive techniques such as mass spec or other techniques?
Yeah. So it's a complicated question about the blood-brain barrier. The answer is probably mostly intact. It's probably mostly intact. There may be some variability in that, but largely I think it's intact. Now, the blood-brain barrier is more robust for preventing things from getting to the brain than it is for preventing things getting from the brain or the spinal fluid into the blood. So all the things that we detect in the spinal fluid probably are there in the blood. They may be there, as you say in your question, inherent to your question, they may be there at very low concentrations. So one problem is we might need to have a very specific test. And we saw this with neurofilament. First, we could only detect it in spinal fluid. Now, with more sensitive assays, we can see it in blood too. There's actually another problem for some molecules, which is that they may be coming from the brain, but also from other parts of the body. So SOD1, for example, which was important for the tofersen program, we needed to measure SOD1 in the spinal fluid because that's what's coming from the central nervous system, from the brain and the spinal cord. You can measure SOD1 in the blood. There's actually tons of it, but it's coming from blood cells, liver cells, lots of things that are not the motor neurons or relevant to the motor neurons. So there are a variety of reasons we have to kind of look in different compartments.
Great. Thank you. I think that was all of the questions. There was one question about a compound known as AKV9, which was formerly known as NU9, that's been developed by Northwestern University. And the question is whether, if we have any information about when it will be in clinical trials. I know they're working on it. I don't know, James, if you know about it. Perhaps we could invite Hande on a webinar, maybe at one of the different organizations' webinars, because we get this question a lot actually on these Thursday webinars. So I just want to make sure we find a way to get more information.
Yeah, I mean, we do work with Hande, and she's wonderful, and so yeah, it would be great to see if she could maybe do a Neil's webinar or another webinar to kind of give an update on where that stands. That's a great idea. Yeah, absolutely. Thank you for the question. Any last comments, James, before we end? I want to come back to this question of, can you drive home after an LP? And I would just say, if you're feeling at all uncertain about it, have someone come with you. It's always good to have someone with you just to give some sort of moral support. I think for all the reassurance we give, we understand that this is, it's not an easy thing to kind of just go through and do, and it can be nerve-wracking. So it's good to have someone there with you. And I would just say, always good to have a friend or a support. Absolutely. And with that, I guess we'll end the webinar for today, but we'll certainly be here next week. Thank you. Thank you so much, James, for coming. Thank you very much. Okay. Bye-bye. Bye-bye.